Hi all, welcome to Anjali's MedSphere. So आज हम पढ़ेंगे electrophysiological studies के बारे में. Why we are studying electrophysiological studies? Have you ever experienced tingling sensation or numbness on your fingers or hands or legs? It might be due to some neurological problem. So let us look into this electrophysiological studies which help us to understand why this is happening with us. So, to give an introduction regarding the electrophysiology study, it is a group of diagnostic studies to rule out how the function of the electrical impulses or electrical activity inside the brain. Like when we do a CT MRI, it will help us to understand whether there is any lesion, any structural abnormality, how the structure, or is there any abnormality in the structure of the brain. But here, it is a specialized diagnostic investigation that will assess the functional integrity of the nervous system that include the peripheral nervous system, the neuromuscular junction, and the muscles. So, we study about all these kinds of tests. We should aware about what is central nervous system, what is peripheral nervous system, and what is neuromuscular junction, and what is muscles. So in central nervous system, it is divided into brain and the spinal cord, isn't it? So brain is having everything. It is the controlling. It has a billions of neurons which controls everything that is happening with our body. Whatever the impulses is generated and it is transported into the other parts of the body. So that is done in the brain. But in the peripheral nervous system, it is again divided into autonomic nervous system, somatic nervous system, or parasympathetic and sympathetic nervous system. So in peripheral nervous system, of all the impulses, all the signals, all the information from the periphery is transported into our brain, and that is how we understand if it is a sensation, touch sensation, or if it is any hot or cold that is experienced on our legs or our hands. So that communication or that signals from here it is transported into the brain. So that is about the peripheral nervous system. Now coming to the neuromuscular junction, there is in neuromuscular junction you can see there will be an axon terminal and there will be a synaptic cleft and then the muscle fiber where the receptors are present. Whenever stimulus is stimulated, that will cross the synaptic cleft and it will the receives or the information is received by the receptors. So whenever an action is stimulated, it will release the calcium channel and ACTHs, acetylcholine receptors are released, and that will pass the synaptic cleft and it will reach the receptors in the muscle fiber, and then the muscle conduction or the contraction of the muscles takes place, and after that the remaining acetylcholine is break down by the acetylcholinesterase, and that results in the relaxation of the muscles. So to in electrophysiological studies, it is a group of diagnostic tests that evaluate the function of the central nervous system, peripheral nervous system, neuromuscular junction, and muscles. So it will help us to understand how the function is, how the electrical impulses are transported from the center to the other parts, or from the other parts to the center of the body, that is the brain. So physiology behind the electrophysiology studies include, like as I said, the electrical impulse. That is, the nervous system communicates through an electrical impulse. That is called action potential. Action potential is the fast or temporary change in the voltage. So at rest it is zero. Whenever it is stimulated, it will sodium channel. So it will the sodium channel is open. Sodium influx occurs and it results in a positive state, that is the depolarization, and then after the entry of the sodium the potassium exits and that is then leads to the repolarization. So that is the main, like, resting membrane potential. Neuron maintain a resting membrane potential of approximately minus 70 megawatt. So it has a sodium potassium pump and selective membrane permeability. So the sodium channel opens, the sodium enters the cell, that is depolarization, the positive state, and the potassium exits the cell, that is the repolarization, and the nerve impulse starts or travel away from the cell body of the axon and it is transported. So this is how the working. So here in electrophysiological studies, we are measuring it. These studies will help us to understand whether this is happening or taking place properly or not. Measures the speed and efficiency of the electrical impulse conducting through the nerve, especially the peripheral nerves. And here, both motor and sensory is assessed. In motor, it is mainly done to detect whether there is any neuropathy, any peripheral neuropathy, or any nerve compression syndrome is there or not. And traumatic brain injury, entrapment neuropathy. So that is when it is indicated. The principles mainly include we are stimulating a peripheral nerve. If it is in the median nerve at the rest, it is stimulated. We will be noting how the sensation is felt in the index finger. Okay. So we are placing some electrodes, whichever the nerve should be monitored. We are placing the electrode and stimulated, and there will be some exaggerated movement activity when there is an abnormality. So the main thing we will be looking into the conduction velocity. It is of normal 50 to 70. Latency, conduction velocity, how fast the impulses is traveling. That is it. Latency, the time taken for the impulse to travel, how long it took to reach the destination. In amplitude, how many neurons are functioning properly. It will be reduced in case of axonal damage. Action study. So interpreting, if there is a demyelinating case, we know that over the axons there is myelin. When in cases of multiple sclerosis and all, demyelination takes place, and that will affect the conduction of the velocity. In electrical impulse, conduction will be affected, so the velocity will be decreased. But in the case of, there will be slowed conduction, prolonged latency, the time will be taken pretty long. Conduction block can be there because of the demyelination. But in the case of axonal neuropathy, inner to the myelin there is axon. So axon means more dangerous it is. So it is in the case of diabetic neuropathy at all. If there will be axonal neuropathy, axon is also involved. Then the loss of neuron is taking place now. So that will show a reduced amplitude. The number of or involvement of the neurons is less, so the wave will be less. So near normal conduction velocity can be there initially. So this is how we will interpret nerve conduction states. Now, electromyography. In electromyography, what happens is the muscles are evaluated, electrical activity at rest, and when we are giving or ask the patient to do some activity or exercises, at that time also they will be evaluating how the muscles are working. If it is not working properly, they will also show some twitching movement or exaggerated movements. So that is about the EMG. At rest and during contraction, we are looking into this. And how the procedure, we are inserting needles into the muscles that should be evaluated at rest. During mild contraction, during maximal contraction, we are monitoring. It also takes 30 to 60 minutes, and in the case of NCS also, it takes 30 to 60 minutes. And evoked potential, as I said earlier, measuring electrical response generated after a sensory stimulation. Like visually we are giving stimulation. Brainstem auditory evoked potential means we are giving, making the patient to hear clicking sounds. Somatosensory evoked potential, touch sensation be involved. And repetitive nerve stimulation means here the neuromuscular junction is evaluated in the case of myasthenia gravis and all. Autonomic function test is means example like heart rate variable. We know that autonomic system is dealing with involuntary muscle movement. Involuntary muscle movements are done by the autonomic nervous system. So like heart rate, and it is not under our control. So into function to test that, heart rate variability means deep and shallow breathing is done, and at that time what change is happening with the heart rate, that is monitored. Valsalva maneuver, we are like, we are blowing up a balloon or a tube against some resistance or force, and what changes is happening with the heart rate. So that is tilt table test from a supine position, a lying position, we are coming to the supine position, and we are monitoring the change in the heart rate. So that is tilt table test. QSART is quantitative pseudomotor axon reflexes. Here this like हमारा जो sweating glands is stimulated, making the patient lie down, which all areas, especially arms and all, acetylcholine related. Capsule is kept over there, and electrical stimulation is given to that. And likewise, the nearby sweating glands are stimulated, and we will look into the humidity, the amount of the sweat. So that is how this quantitative pseudomotor axon reflex test is done in autonomic function test. So it is used to for indications such as diabetic neuropathy, Parkinson's disease, and dysautonomia. So electroencephalogram, activity of the brain recording, and especially in the case of seizures and epilepsy, sleep disorders. So what all advantages and disadvantages with this is, it is a non-invasive type of testers, and there is no need of any NPO or fasting status. But the limitation is operator is needed. Some procedure, especially EMG, need a needle insertion, and that time you have to monitor whether the patient is taking any anticoagulants or any implanted, pacemaker is there or not. So that is about electrophysiological studies. Hope you got some kind of idea regarding it. Please watch the video, and if you like my video, please like, subscribe, and comment for any queries. Thank you so much. Let us see soon in another video. Thank you.